All right, so I wanted to first off just thank you both. I'm really excited, that we can have this conversation. I wanted to just start by having you both introduce yourselves and maybe give a little bit of your background, so that people know why it was you I approached to have this conversation. So maybe Jim Summers, if we could start with you.
Yeah, I'm Jim Summers. I retired a year ago from AbbVie after spending my whole career, over 30 years at Abbott and AbbVie, in drug discovery research. I'm originally trained as a chemist, PhD in chemistry from Harvard, and then over the years became more and more involved in biology and all the other multiple aligned disciplines that are involved in the development of drugs. I've worked in a variety of different areas in drugs for immunological diseases, oncology, and for neuroscience diseases. Have never actually, though, worked in antivirals, but I think the learning that has taken place across all those diseases, there are many common themes that I can offer some insight into viruses as well.
Awesome. Jim Stolzenbach.
Hi, Elizabeth. Yeah, thank you very much for the opportunity. It's great to be able to talk to you and Jim about this. So I'm a trained pharmacologist, a PhD scientist. I've been in the pharmaceutical industry for over 30 years, primarily focused in cardiovascular and renal disease. Later in my career, more into immunology. So I have a broad background in development, primarily in development of drugs, both biological and small molecules. And in clinical trials that have focused on all kinds of disease states and really the entire life cycle. So I've worked in drug development all the way from trying to translate early discovery studies into humans, and then the large trials that are required for actually getting regulatory approval across the world. So I'm retired at the moment. I'm consulting and enjoying working with a lot of different companies, mostly smaller companies, on trying to bring new therapies into clinical development. And so this is an area that I'm really very interested in and very, very happy that I can talk to you about.
Well, and both of you are people who've had a lot to do with my own education in pharmaceutical science and in science in general. So I think I credit both of you with a lot of my interest in science. So I'm excited we can have this conversation and hopefully just help people understand a little bit about what's going on right now in the world of science as it relates to COVID-19. Right? There's so much discussion, so many things you can read, so many comments that you hear from from our government leadership about all that's going on in terms of vaccine development and development of medications that may potentially treat COVID. And so what I thought we could do was really just kind of back up a little bit because, we all know COVID-19 now, but, we know that it's a coronavirus and we know that it's a virus. Maybe we start there and talk about what is a virus, and what do we know about this? And what does that tell us about where we need to be directionally looking for answers? So maybe Dr. Stolzenbach, we can start with you.
Sure. Yeah, I'll give it a shot. A virus is basically a parasite. So it's a very small parasite that requires other cells in order to replicate and to survive. So it has to infect another cell to be able to survive, really, in the environment. And viruses can attack people, they attack animals, they attack plants. They're even very small. They're smaller than bacteria, so viruses attack bacteria as well. The key for them is to get into the cell and use a cell to replicate itself. And that's what they do. So when they enter the human body, for example, they have a process for getting into a cell, and then they use the cell's machinery to reproduce themselves. And they make millions and millions of copies of themselves that spread throughout the body, or the plant, or the whatever it is that they've infected. So viruses are ubiquitous. They're everywhere, and they can infect again, animals, people, plants, all sorts of things. A coronavirus in particular, well, maybe before I get to a coronavirus, viruses are made up of a genetic material, DNA, which most people are familiar with, or another genetic material, RNA, which is also present in all cells. And some viruses have DNA, some have RNA. They all have protein that surround the genetic material, and some of them have some fat or a lipid that helps protect the virus from the environment. So that's how viruses survive on inert surfaces. They are protected by this protein coat. A coronavirus, corona means crown, and a coronavirus has these spikes. So when you see the pictures of coronavirus, they show a ball with all these spikes on it. That's one of the identifying characteristics of a coronavirus. They have these protein spikes, and those spikes are actually indicative of the way that the virus attacks a cell. So that is one of the primary characteristics of a coronavirus, those protein spikes. COVID-19 got its name because it's a coronavirus, it's coronavirus disease, and it was discovered in 2019. So it's COVID-19 is the short name for coronavirus disease 2019, basically.
Okay.
Maybe I could just build on that a little bit and talk about the fact that the viruses are collected into families, just like other life forms, animals and plants are. You know, tigers, lions, house cats are all feline species, and the same is true of viruses. They can be divided up in the same way. And there are multiple coronaviruses, not just COVID-19, as Jim was saying. In fact, the common cold is a coronavirus. And there are other coronaviruses that infect animals. And many of these obviously are, in the case of the animal viruses, don't affect humans at all. Common cold is a little bit more modest in its effect. We're over in a week or two. SARS was another coronavirus that was actually more serious, that disappeared a couple decades ago now. And now the latest to emerge is COVID-19. So this is all part of this family taxonomy that makes up viruses.
So what do we know then? I mean, obviously it's the fact that you're able to describe already what this virus looks like, right? Having the spikes on it, and the fact that it's part of this family of viruses, and some of them pretty well known to us already. What do we know about the way that this virus works specifically in the body? Maybe Dr. Summers, you can take that one.
Sure. I think it's been really exciting to see how quickly we've been able to, the scientific community that is, has been able to try to understand this virus. Just a few weeks after it was initially appeared in China, the genetic instructions, the genetic material that Jim Stolzenbach talked about, was completely sequenced and understood and available. Now, that's something just a decade or two ago would have taken months, if not years, to be able to do. And this happened within just a few weeks. Now, in addition to that, there were other understandings. The pictures that we see all the time of the coronavirus with the spikes that Jim talked about, that came out within a week or two. So all of those, both the advances in science that have occurred over the last decade or two, together with the interest of the broad interest across the entire scientific community, has made it possible to make really rapid progress. I think also the point that Jim was making, that this is part of a family of viruses that we have some experience with from previous versions, SARS, for example, or the common cold. That enables scientists to make an analogy between those old viruses and this new one, and predict a little bit better about how it's going to behave, how it's going to be spread, how people are going to develop immunity. So we have a basis for understanding. It's like, to use my analogy before, if we'd studied tigers, we'd have a better understanding of lions. That's being in the same family.
Yeah, I agree with that, Jim. I mean, we've really come a long way very quickly, and we know specifically how the virus gets into human cells, what receptor, what protein the virus attaches to. And one of the interesting things about this virus is that it attacks a protein that's very common on a lot of different human tissues or organs. So one of the reasons why the virus is as dangerous as it is because of the fact that it can attack a lot of different cells within the human body. And to Jim's point, this information has been generated extremely quickly. And it's really important to understand how the virus gets into human cells, because if we know that, we can figure out ways to block that and to stop it. And that's really the focus of where coming up with a vaccine or a drug to treat the disease is going to come from. So that basic scientific knowledge, which in this case has been accrued extremely quickly. I mean, this is really very rapid. In the space of a few months, the world's scientific community has been communicating with each other all of this various information. So it's a little overwhelming because you hear all these different things and it comes from all sorts of different places. But I think it's comforting to think that the scientific community anyway knows how to access this kind of information and put it into ways that can be used, and utilized in a way that will really help everybody over the long run. But from a layman's perspective, it's just an avalanche of information coming at you from all over the place. And I think the focus is, it's important to understand that all of this information is being generated very quickly, even from experienced people like Jim and I. It's amazing how fast this is happening. And it speaks to the rapid development of technology, which all of us have invested in. All of the work that we do to pay scientists in the US and invest in the NIH and other things is really the reason why we've been able to approach this as quickly as we have. So.
Well, so you bring up a great point then. So there's all sorts of information now about how the virus works in the body, what it attacks, what, like you said, the variety of cell types that it can attack in the body. So then where do you start in terms of thinking about how do you treat this or how do you vaccinate against this? What are those initial questions that you have to look at and ask yourself before you really start going down any particular pathways? And maybe Jim Summers, we start with you on that one.
Yeah, let me talk about three different approaches that I think are important to think about, all of which I think have been in the press recently, and try to provide a little color and background to those. The first and probably the most important is our vaccines. And vaccines, of course, are a way of being able to teach the body's immune system that a particular invader, in this case COVID-19, is dangerous and needs to be developed. So it's a way of teaching the body to develop antibodies to the COVID-19 virus. And this is really the best approach, because it's something that all of us can have walking around that provides us protection against the virus, and we don't have to take anything for it after we've been vaccinated, maybe boosted. It's always there, and that's why it would enable us as a society, I think, to substantially suppress or eliminate these infections. The second approach to think about is related to the vaccine, using a very similar kind of concept, but it's the use of using people's plasma, blood components from people who've already been infected with the virus and who've recovered, but transfusing that into people who are at risk and have not yet had or have early in their infection. The idea there is that rather than trying to induce the patient to produce an antibody like a vaccine does, you're actually just transferring antibodies from people who've already produced it themselves. So it's more of an active process of being able to give that. That provides a much shorter term treatment, because those antibodies will go away that you've given from somebody else. You haven't taught the body to make new antibodies like you do with a vaccine, but that sort of active immunization through transfusing blood components from other people would be the second approach. And I think that's already being tried. It will be difficult to do that on large scale, because there just isn't enough blood and plasma from infected people to be able to give to millions of people. But in high-risk individuals, like healthcare workers or elderly people or people with other pre-existing conditions, or people who have already become symptomatic, that would be a potential therapy that would be available right away and could go on. The third approach would be more conventional pharmaceuticals, more conventional drugs, antiviral drugs. And to be able to develop those will take much more time. To do that, you would begin by looking at the process by which the virus reproduces itself and lives, the viral life cycle. And you think about a virus, what it must do to reproduce. Jim talked about this earlier. It has to attach to a human cell. It has to be able to get inside that cell. It has to open up and spill out its genetic material. It has to trick the cell into producing copies of itself. It has to evade the cell's natural defense system. And if we can understand each of those various steps along the way and how the virus does that, then that represents a potential point to develop a drug to intervene. So for example, if we could find a way to block the ability of the virus to attach to a cell, that would pretty much stop the process of viral replication. So those are the kinds of things that people are looking at today. Those will take longer. The drug development of that sort may take a few years. That obviously would not be beneficial for COVID-19, but it could be something that's going to be very useful across other viruses. And the reality is that we're going to have other viruses emerge over time, hopefully not as bad as COVID-19, but if they do, our work to develop antiviral approaches today for this current pandemic will be applicable to future pandemics as well.
Mhm.
Yeah, so I would just add a couple comments to where Jim's been going, and it's a great overview of the ways to approach this. I think one of the things that's happening right now is people are asking a lot of questions, and I would just point out some of the questions that need to be answered in some of the studies and some of the ways that Jim's talking about. First of all, these viruses do change over time, and that can be an issue. The mutation rate of this virus is really not known very well, so that's a potential question that has to be answered. Whether a vaccine will work in terms of producing antibodies that actually immunizes you from the disease is another question. And so I think there's been a lot of questions in the press lately about if you have the disease once, can you get it again? And as Jim mentioned, coronaviruses are fairly common. You can get colds multiple times. For this particular virus, it's not been proven that once you have it, you cannot get it again. So even if a vaccine generates antibodies, how effective those antibodies will be and how long that immunity will last, if there is immunity, is a question that we really don't know the answer to. So there's a lot of ways to approach this. There's a lot of different drugs that are being looked at. There's a lot of different vaccine approaches that are being looked at, but this virus is very complicated. And so it speaks to the requirement to really take a lot of different approaches at this. Go after a vaccine, go after a drug or a biologic that can work to help ameliorate the disease, if you have it over time. But there's no firm direction that we know is going to work right now. And so we have to take these multiple approaches. And again, it's confusing because you see a new thing coming out every day. Some of these approaches are still not in humans. There's many human trials going on. But the reason for that is because we can't be certain which one of these approaches will work. And so there are some viruses that we've never been able to develop vaccines for, such as HIV. There are others that we have very good vaccines for. There's some like the common flu. We have vaccines, they work reasonably well, but they're not perfect. So you really want to have a wide variety of approaches to this. You want to have a drug that works. So if you get sick, you can be hopefully cured or have the symptomology be less than it would be otherwise. You want to have a vaccine so you can protect as many people as possible. But it's really necessary to have multiple approaches on this. And hopefully a few of them will actually work. And then you can really start to, I think, ease the concern that if you get sick, oh, there's a drug that can help me with that. Or I'll never get sick because I have a vaccine here that, I can take and will protect me. So we're a ways away from getting there, but the good news is there's a lot of different approaches being tried, and there's a very good chance that some of them will work. And that's really, I think, important to say.
I'm really actually quite optimistic because there are so many different approaches, and because such a large fraction of the scientific community is really focused on trying to do this, that something's going to emerge. The other point I think to make, and Jim spoke about the challenges of trying to have vaccines. Sometimes you can have a highly effective one, sometimes it's difficult. There are coronaviruses that infect animals, and farm animals in particular. And there have been vaccines developed to treat those kinds of coronaviruses so that the animals, the farm animals, don't develop the infections. So that tells us at least it is possible to develop vaccines that are effective against coronaviruses.
So that actually raises another question. It made me think not only in the discussion about vaccines, but then you talked about animals too. And it made me think about this concept of herd immunity that we've been hearing a lot about. I mean, obviously people are really anxious to get back to some sense of normalcy and getting back out in groups again, et cetera. But a lot of the caution is we've really got to make sure that there's herd immunity, or, that we have some of these things in place, like you mentioned. Can one of you just address this idea of what is herd immunity? How should we be thinking about this?
Yeah, let me start, and Jim, you please jump in anytime. So the concept here is that there are people that, for whatever reason, may never be able to get immunity to a virus. So they're either very sick, the vaccine won't work, their immune systems are compromised. There's other reasons why a certain number of people in the world will never be able to be immune to the virus. So the concept of herd immunity is if you have enough people in the population that are immune to the virus, that it will basically protect the small percentage of people that cannot become immune to it. So the concept is to get as many people exposed to the virus, either through a vaccine or through getting sick, as possible, so they build up an immunity to it, and therefore the virus can't transmit, it can't survive. Again, a virus has to have a host to live in. It's like a parasite, right? So if enough people are immune to it, it can't infect anybody, and so it kind of goes away. This virus is very, very infectious, so it's very hard to prevent people from getting it, and that's one of the issues that we face. But for herd immunity, you need a large percentage of the population to be immune to it, at least 70 to 90%, in order to make the chance of the virus finding a person that's not immune to it very, very low. For vaccines, the standard is you really want to get about 95% of the population vaccinated, so that there's only a very small percentage of people that are not immune to it. And again, it just makes it very hard for the virus to find things to attack. If everybody's immune to it, it kind of dies out. There'll always be hot spots where it kind of survives because there's a fair number of people that are not immune to it for whatever reason. They can't get the vaccine or whatever. So these things tend to pop up occasionally, but if you can get enough of the population exposed to the virus, either through a vaccine or through being sick, again, you're protecting the people that for whatever reason cannot become immune to it. One of the things that I think is hard about this virus is, even though we talk about the huge numbers of people infected, the percentage of the population that's been exposed is still pretty small. It's probably in the 10, 20% range or something like that. And again, for herd immunity to kick in, you really need to have very high numbers of exposures. 70, 90% is really where you start to get good, strong herd immunity. And again, the concept is make it very hard for the virus to find people that it can infect. And by doing that, the virus will die away. If it doesn't have a host, it's not going to be there. Now, these coronaviruses, of course, exist in animals. That's probably where this one came from originally. So it's very hard to eliminate them completely. But if you can make most of the human population immune to it, you can prevent the massive transmission that's occurring currently, because no one had ever seen this virus before. No one had immunity to it. So it's very easy for the virus to find people to infect.
There's an interesting irony to our, an unfortunate irony, I think, to our current public health measures that we're taking to try to control the virus spread. And that is by staying in at home as we are. And I'm certainly advocating very strongly we continue to social distance and stay at home and try to avoid spreading the virus to other people. But as we do that, we make it harder to obtain herd immunity because we're not infecting enough people to develop that. That's why a vaccine becomes the best approach. We can't really expect that herd immunity will arise simply by exposing people in the community through the normal transmission of the virus.
Yeah, that makes sense. Thank you. So one other question I would just ask on the side of we were talking about, I think it was Dr. Summers, I think you mentioned, there are something like 50 clinical trials going on, looking at different vaccines. I know that there are a number of different trials going on as well for potential medicines, potential treatments, some that have been used in other diseases, and maybe there's some thought that they could have a good effect here, and others that are novel, that are new treatments. Maybe, Dr. Stolzenbach, can you talk a little bit about clinical trials, and I guess what we'd be looking for here, in being able to say something is something that we would potentially see getting approved. What would you be looking for coming out of clinical trials?
Yeah, no, that's a great question. So there's a couple things that I think are really important to say. First of all, development for vaccines and drugs is different. There's different approaches to it, but conceptually, they're very much the same. And so the first absolute number one priority before you do anything is to figure out whether or not you can safely give an agent to a person. So there's always some risk the first time a new vaccine or new drug goes into a human. There's always a risk of causing harm, which of course you don't want to do. So the first priority is safety. And usually in development of a drug or a vaccine, the way we do that is we introduce I'll use a drug as an example, we introduce the drug to a small number of either healthy people that will take the drug, and then we can figure out, does the drug cause side effects? How does the drug work in the body? How long does it last in the body? Those kind of things. So we can actually measure the drug in the body and say, okay, it lasts a long time. That's a good thing because we want to be able to treat people. It doesn't cause, any serious problems, but you look at that in small numbers of patients first. So what we call phase 1, you'll hear development, is usually 50 to 100 people. And you give the drug at relatively small levels and you slowly build up. So there's several questions after safety that we want to ask. So one, is it safe? And then number 2 is, how do you give the drug so it can be effective? So what dose? How much drug do you give to a person? How often do you give it? Is it safe over long periods of time, or do you have to give it for a short period of time? So there's a lot of questions that you have to answer. The other question that always comes up is it safe in different populations of people? So is it safe in women? Is it safe in men? Is it safe in African Americans? Is it safe in all the different populations you can think of? Now, you can't answer all those questions at once. So again, the first stage is it safe? You look at small numbers of people, and then you try to understand the drug as best you can. How long does it act? How long does it stay in the body? What are potential side effects? Things like that. The next stage of development is to introduce it to more people, what we call phase 2 development. And that really looks at maybe 100 to 1,000 patients, and these are almost all patients now. We're moving out of healthy people. And the idea here is to say, does this drug work? Does it show some efficacy against the disease? And you might be looking at a marker, an indication that it might work, something that tells you that the drug is actually doing what you want it to do. You're looking at symptoms in people. Does it relieve pain? Does it make your joints move better? You're measuring people's response to the drug. And you're trying to get an idea, again, in what populations might the drug work. So very important question. For example, in this virus, does a drug work if you give it only early, at the sign of the first symptom, or can it work later on? Another question, is it safe to give to people in intensive care units? So now you have a patient that's on a ventilator, they're taking all kinds of other medicines to try to support their life. Can you give our new drug in that kind of a situation? So you have to look at a fairly large number of people at various stages of the disease. And coronavirus, very early, you have a sneeze, a temperature increase. Okay, let's give the drug now, right away, to try to prevent it from getting any worse. Does it work there? Some drugs might, some it might be better later on. The body's response to the virus is extremely complicated. And actually, one of the problems that we have is that people's immune systems actually over-respond to the drug. So the point I'm trying to make here, there's a lot of things you have to consider as you're developing these drugs, and you start to look at that efficacy in those phase 2 trials. Then in phase 3, you take large numbers of patients. Now we're talking about thousands of patients. And now you're really looking at all the different subsets of people that I talked about. Is it safe in women? Is it safe in men? Is it safe in African Americans? Is it safe when you're in an ICU? You're really looking at a large number of questions that you're trying to answer. So in traditional drug development, you kind of go through these stages in a sequential way. You look at it very carefully, then you write up a big report and you send it into a regulatory agency, and they take a year or more to approve it. And so that's why drug development is very expensive. There's a lot of questions that we have to ask to prove that this can be a safe and effective treatment. For a vaccine, it's amplified by the fact you're giving the vaccine to healthy people. You're trying to prevent them from getting infected. And so the question around safety is even more important. You really have to understand that the vaccine is safe. And then again, all the same questions about efficacy are there. What dose of the vaccine? How often do you have to give it? With a vaccine, one of the critical questions is how long does the immunity last once you give it? So the point here is that there's a lot of complicated questions to ask. That's why drug development normally, when you get into the clinic, the very first person to an approval is usually, 5, 6, 7 years, something like that. It takes a long time to answer all these questions. There are cases, and oncology is a perfect example, where a very serious disease, people are dying. We do everything we can, and the regulators do everything that they can to accelerate that process. So you look at smaller numbers of people, you take more risk, you do things in parallel, and so you can really do a lot of things to move through those drug development phases very, very quickly. But I would just leave everybody with the thought that the first priority is safety. Can this be given safely? And then secondly, how does it work? And then, is that true in all the different kinds of people that you can think of? That's really critical. For drugs that are already on the market, and we've heard about some drugs that are being repurposed, in other words, it already exists, boy, will it work in this case. There, the question is, it's been approved for one disease. Can you use it in another disease? And is it safe in those patients? And is it safe under the conditions that you need to give it to? And is it safe at the same doses? Does it work at the same doses? So repurposed drugs you can use more quickly, potentially, but the question around safety and efficacy still exists and needs to be answered.
Just to build on a couple of points, I think Jim did a really nice job of taking you through the whole drug development process there, and maybe a couple of points specific to vaccines. We hear a lot about the idea that it might take 12 to 18 months to develop a new vaccine for coronavirus. And I think Jim and I both feel that's incredibly fast. And for the reasons Jim just told you, that this is normally takes multiple years to be able to do. So first of all, what makes it specifically long in this case for vaccine development? And what's being done to try to make it faster than the usual. So I think from what makes it long for vaccines, there's a couple of points. Jim talked a lot about safety. If I give a volunteer or a patient a vaccine today, I want to make sure that 6 months from now that there isn't something untoward that develops in that patient that might not have picked up right away. So while it might be tempting to say, boy, this drug looks safe, because after a week or two, let's start giving it to everybody, I think you have to be more careful about the long-term safety, and that's one issue there. The other thing that makes it long in this particular case is the way that the trials need to be done. You could envision maybe a trial that would be really fast, which you would say, we'll give 100 people a vaccine, and we'll give 100 people a placebo, and we'll expose them all to the virus and see whether there's more people who get the infection in the placebo group than get it in the vaccine-treated patients. That could probably be done in a couple of weeks. The problem, of course, is no one should be exposed to the virus intentionally. It's too dangerous. It's just unethical to do that. So what we have to do instead is to give patients or volunteers the vaccine and then allow them to go about their normal daily life. Over the course of months, there's a probability that some of them might be exposed to the virus, and we look for a difference between those that received the placebo and those that received the vaccine. But that requires much more people and much longer time to be able to see that develop, which is one of the challenges. Now, this is all happening very fast for a number of reasons. One is the huge effort that's going into it, which is really understands the urgency in trying to figure out ways to make it as fast as possible. I think the other thing that's happening is everyone involved, including the regulatory agencies like the FDA, are willing to take on a little bit more creativity and a little more risk than they would normally in this situation in order to bring the vaccines or the drugs to people more quickly. This, as I say, has some risk associated with it, but the benefit is huge in the chance to be able to provide a treatment or a preventative treatment like a vaccine to patients.
Yeah, the other thing I'll just say about the timelines, we haven't talked about this, but for vaccines in particular, some of these are very complicated biologic products, and so they're very expensive to make. In traditional drug development, a single company would not take the risk to build a facility, which might cost $500 million to manufacture a vaccine until they know it works. So they would wait till those larger trials that I was talking about, those big phase 3 trials, to actually show a benefit before they would spend the money to build a huge facility to produce it. Because for a vaccine, especially with this virus, we're gonna need hundreds of millions of doses. So it's going to be you're going to need a facility or multiple facilities, which is a huge investment in time and money. I think, again, as Jim spoke to, the regulators, they're willing to do a lot of things to help move things along faster. This is a place where it's very encouraging to see that governments around the world and various nonprofit organizations are actually stepping up and saying, we're willing to help fund at risk making manufacturing facilities available, so that we're going to pick one or two of the 100 choices that we have, and we're going to bet that that's going to work, and we're going to start making a factory now so that as soon as we know it works, we can start producing drugs. Now, there's a huge risk there because we don't know which one of these is going to work. We could build a factory for something that never actually gets produced. So it's a global effort to really minimize the risk. And one of the things that's happening is that, usually pharmaceutical companies are competitive with each other. Governments, are reluctant to step in and support pharmaceutical development because it's a profitable industry on its own. Here, what we're seeing is many, many companies joining together to do everything they can collectively to spread the risk among the industry. And we see governments throughout the world willing to help with mitigating some of the risks, so that if a company makes a bet and it's wrong, they're not going to go bankrupt by doing that. And so, I think the seriousness of this disease is visible to everybody, and there's a lot of organizations stepping up, regulatory agencies, government agencies, and the pharmaceutical industry as a whole, to make this work in ways that would never work under normal circumstances. It's just not economically feasible. So you can shave easily 6-8 months, almost a year, just by the manufacturing process alone.
So that's, I think both of you sound really optimistic, really encouraged by what you're seeing, as scientists, and how quickly we've been able to share knowledge and really kind of join forces against this. So I guess my last question to you would really just be, what would be your message to people about all of this, with so much information coming at them and so many different sources of information, not always knowing, what to believe, or maybe it's not even all understandable to a lot of us lay people. What would you what would you say? What would be your kind of parting thoughts? Why don't we start with you, Dr. Summers?
Well, I would say, first of all, I would ask everybody to continue to do what they're doing at home right now to try to stay safe. A big part of all the public health measures that have been put in place, the social distancing, the mask, and so on, is about trying to buy time. And so trying to allow the curve to be flattened and spread out a bit longer, so that there is a time when a vaccine can be available or another type of antiviral therapy can be available. So I think it's really important for all of us to be patient with that, to try to listen carefully to our leaders and the experts about what is being recommended. And even though we're all, certainly I know all three of us on this call here are anxious to get out and about with our normal lives, recognize that what value we're really doing by doing that and giving the researchers the time they need to be able to develop these new approaches.
Yeah, no, I agree with Jim totally. I would just say a couple things. I think one is I would urge everyone to exercise what I call critical thinking. So every single thing that you hear is not true or may be true or whatever. So go to replicable, to sources that you can trust. I'm a scientist, but there's a lot of sources, the NIH, other sources, Johns Hopkins University, a lot of these very good fundamental academic and scientific organizations, the American Association for the Advancement of Science, they produce Science Magazine. All of these places have websites that will approach this information from a layman's perspective. So rather than trust your favorite newsman or cable network, or Facebook page, try to go to some sources. And if information is replicated in multiple sources, then it's probably something that you can believe in. And that critical thinking part, is this too good to be true, or too crazy to believe? Think about that a little bit. I think Jim and I think we're very optimistic that something will come of this, but it is going to take time, and everybody's situation is not the same. So I would urge everyone, even when the restrictions are lifted throughout the country, and they need to be, of course, at some level, everyone needs to take their own individual risk into consideration. If you're older and have underlying medical conditions, et cetera, you need to be more cautious about what you're doing until there is a very good treatment or a vaccine, and that might take some time. So your own personal safety, you really need to pay attention to. But I think the effort that's being done here by the world is amazing. We've made a lot of mistakes in this. I think it would be crazy to say that everything has been done perfectly. And I guess my one hope is that we all can kind of get together and learn from what we've experienced here and make it better next time, because there will be another virus that will pop up. It's inevitable. And we need to be better prepared for it than we were this time around as a world, because we're so connected that you can't isolate yourself from from any of these things anymore.
Well, thank you both. I mean, I can't thank you both enough. I joke that this is like getting the band back together because we haven't done this in a while. But to be able to talk to you both is just it's been great. You know, for me, I love hearing your perspective. You've always made it so understandable. You know, to someone like me, to be able to get passionate and excited about science, and you know, certainly from what I'm hearing, you know, I feel really optimistic about where things are going as well, and all the collaboration taking place across so many sectors. So thank you both. Thank you on behalf of Father Ricardo and the parishioners who have been excited to hear, you know, more about what's happening. And I hope that you and your families continue to stay safe, and that at some point maybe we can get together for lunch. Wouldn't that be nice?
Absolutely.
Thank you very much, Elizabeth. We appreciate the opportunity. Stay safe.
Thank you very, very much.